So let's look at what we have here. So we have a reaction, Q going to R. And so the first part of that problem, 21A. So if the equilibrium constant is greater than 1 and equal amounts of Q and R are mixed together, the reaction will generate more what? And as you can see there, the answers are. So just quickly for 21A, remember equilibrium constant, KEQ equals products over reactants. For us, that's R over Q. So if it's greater than 1 and we start with the same amount of Q and R, therefore we're going to make more R, because if they're the same, if Q equals R, that means currently when you do R divided by Q, that is 1. And so the way to make that greater than 1 is to add more R. Then if it's between 0 and 1, then it's just reverse. So 21B then, if KEQ equals 14 and the starting concentrations of Q and R are both 34, then at equilibrium, what will Q be and what will R be? All right, so KEQ equals the concentration of R divided by the concentration of Q. And we're saying that is equal to 14. And we are also saying that they both start at 34. So that's just like what happened in A, right? We're saying that Q equals R, so currently the ratio is 1. And it needs to be greater than 1, which means if we start at 34 for R, we have to add some amount of R. So I'm going to say that's plus X. And for Q, we're going to lose the same amount of Q that is given to R. Because if we look at the ratio, every 1 molecule of Q makes 1 molecule of R. So R is going to gain some amount, so plus X. Q is going to lose that same amount, minus X. And then you just solve for X and you add them back here. And so if KEQ is 14, then Q, the final concentration will be 4.53. R will be 63.5. So that's how you go and solve 21B. And excuse me for just 1 second. I'm going to let my dog out before she starts making a noise in my room. All right. So with that, let's get to our actual material. Just really quick, I want you to know. All right, if you could, yeah, send me an email for if you have to miss class because of some appointment, just send me an email. That would work just great. All right. So we have a little bit to cover from Monday. Just to remind you what we were talking about, we were looking at DNA, and we talked about how to sequence short DNA strands. And the idea is, let's say, you either have a very long DNA or you just have a short DNA. If you have a very long DNA and you want to sequence it, one of the things you can do is that you can use the restriction endonuclease to cut that DNA and then sequence it using the Chain-Terminator sequence method. That's how we sequence large amounts of DNA in the past. Now, the Chain-Terminator sequence method is really just used for sequencing small DNA, 1,000 kilobases, not that big of a sequence, but it's still used today because it is fast and cheap, and it's good for a lot of laboratory experiments. So that's what we ended on Monday, talking about the Chain-Terminator sequence. And I showed you this image of what a real output from the Chain-Terminator sequence method looks like. And before we get into new material, before I forget, test 1, where's my pen? Where'd that go? Okay, test 1. So this 10-week class, so it's very condensed. So test 1 is next week, Wednesday, and we'll most likely start that at 9 a.m. and you'll have probably like 11 a.m. I'll probably give you some leeway, 1 hour to take it. Yeah, I'll talk more about that on Monday though, as I'm probably going to work on it over the weekend here. But just be aware, our test 1 is coming up fast. All right. So what we're going to talk about to begin with here is how do we actually sequence whole genomes, right? The Chain-Terminator sequence method, like I just said, you're looking at 600 to 1,000 kilobases, while, a human has millions of kilobases. So using Chain-Terminator sequence on that and on a whole human genome, not practical. And so during the Human Genome Project back in the early 2000s, different techniques were developed and are still being developed today to sequence whole genomes. And so we're going to go over 3 different methods here. The first method is called pyrosequencing, also called 454. And what you do is that you take an organism's DNA and you break it into pieces. So you take DNA and expose it to restriction endonucleases to break it apart. Once the DNA is broken apart, you attach it to plastic beads. And it's on these plastic beads that we are going to try and read the sequence. To do that, let's start at our picture on the bottom left here. Get my tablet over here. So on the bottom left, we have our plastic bead, that's this blue thing, and the long gray thing is our DNA. And what you do is you add 1 nucleotide at a time. So here, we're adding only thymine. And if our sequence begins with A, then thymine will attach. So on the right here is what our actual DNA sequence is that we're trying to sequence. So you can see that we have 2 A's to begin with, and so 2 T's stick on. We hit it with light, and the nucleotides will fluoresce. And you can measure the fluorescence to know if 1 nucleotide was there, 2 nucleotides were there, 3 nucleotides were there, so on and so forth. So we do that with our T's. Then we wash all the thymines away. And then we go on to our next nucleotide, A. And if there's a T in the next sequence, we would have an adenine stick. But you can see in our actual sequence, there's a C there. So A does not hydrogen bond with C, so no A stick. We hit it with light, we get no signal back, which means no A stuck. So then we wash away all the A's. Next nucleotide is G. Well, there is 1 C there, so we get 1 G to stick. Hit it with light, get a signal out. Wash away all the G's. We then add C's, see if any stick. There was no C's that are hydrogen bonding here, so we don't get a signal out. And then you just repeat. And so you can see this can be a very automated process where when you get your DNA stuck on plastic beads, you just do this loop of adding one nucleotide mixture at a time, measuring the light. You can do a lot of plastic beads at the same time. And like it says there on the slide, 1 million templates you can sequence at once. So you can sequence 1 million DNA strands, which when you do the math, comes out to be roughly 1,000 times faster than the Chain-Terminator sequence method. So that is one way you can sequence a whole genome incredibly fast by using pyrosequencing. Any questions about our pyrosequencing there? All right. The next type of sequencing we're going to look at is Illumina. And this is very similar to pyrosequencing in that you take your DNA, which again is gray, and you break it into small pieces. Then you put those pieces on a piece of glass. The big difference between pyrosequencing and Illumina sequencing is that in Illumina sequencing, you add all nucleotides at once. So all 4 are added at once. And each nucleotide gives off a different signal once it binds. So for example, the way this little cartoon is colored, T would be green, G is blue, C is red, A is yellow. And through some kind of secret magic that Illumina doesn't tell us how it's done, they have engineered their nucleotides to only ever attach one at a time. So you can see we have the same DNA sequence to begin with. It starts with 2 A's. And in the pyrosequencing that we just looked at, when we started with 2 A's, 2 T's were added. However, in Illumina, we only add 1 nucleotide at a time. So we add all 4 of our nucleotides, 1 T hydrogen bonds to our template, hit it with light, we get green light out. Then you wash everything away, reset the reaction, add 4 nucleotides, 1 T sticks on again, hit it with light, you get another green light out, wash everything away, add all 4 nucleotides again. Now a G sticks and you hit it with light and get blue out. So similar to pyrosequencing, but some key differences being that you add all 4 nucleotides at the same time, and only 1 nucleotide will ever hydrogen bond to your template. For this Illumina sequencing, you're looking at a range of 30 to 300 nucleotides at a time. So very short DNA sequences, but you can do many reactions at the same time. So you can do 3 billion reads per run. So you can do 3 billion nucleotide strands at one time. So imagine you're doing a genome. You just cut it up, put those pieces on glass, do this, and you're done. You just have to put the pieces back together and you have a whole sequence, whole genome sequence right there. So that's Illumina. And through these next-gen sequencing techniques, we have sequenced a lot of different organisms. Here on the left is just an example of some of the organisms we have sequenced, you know, humans, mouse, chicken, zebrafish, yeast, parasites. And through the Human Genome Project, where we sequence humans, we found some very interesting things, which is shown in these pie charts here. And one of the interesting things is that the DNA of humans, only 1.5% of it encodes for protons, which is very surprising because, or not protons, proteins. And it's very surprising because proteins are the workhorses of the cell. Proteins do everything for us. They do our chemical reactions. They also help us structurally. They are very key for a cell to live. Yet only 1.5% of that, of our whole DNA, is made to make proteins. 25% are introns, things that we cut out that don't make it into the final product. And I'm not going to go over all the things that are labeled here, but this next pie chart is we're looking at what makes up the 1.5% of our proteins. And it always strikes me as funny that like 40%, we don't know what it does. There's a lot to still understand about the human genome. Enzymes, something that you'd think makes up a lot of your protein coding genes, because a lot of proteins are enzymes, that's only 16.2%. You can see that if you just look over here, you got a lot of structural proteins, membrane proteins, immune proteins, extracellular structure. So just the take home from this image is that the DNA of humans, most of it doesn't make proteins. These images here on the right are just showing what a small amount of change in DNA can do to an organism. So here is 2 different types of corn. Just a small amount of change in your DNA goes from like wild type corn to the corn we eat. The same things between humans and chimpanzees. Our DNA isn't that different percentage wise, but the actual look of a human and chimpanzee are quite different. One other cool thing to notice about sequencing genomes, if you look at like the number of chromosomes, that doesn't tell you complexity of an organism. A chicken has 40 chromosomes. We only have 23. Most people would say a human is more complex than a chicken though. Also, the amount of DNA doesn't actually tell you complexity either. A lot of like mushrooms and plants have more DNA than us. But again, humans would probably say they're more complex. And if you'd like to get your genome sequence, I think prices right now are about $1,000. You get your results back in like a day. So we've gotten very good at doing that. And what did we learn through doing sequences of a lot of different humans? One of the key things that sequencing can do for us is to help us understand diseases. By looking at individuals who have inherited diseases versus individuals who don't, we can start comparing their DNA and see where mutations are. And we have found some mutations like cystic fibrosis, where you have a mutation in one gene that causes this disease. However, that's actually very rare. Diseases are usually many different genes that are being mutated. So it's very hard to pinpoint, that this mutation will lead to this gene. There are some cases where that happens, but it's rare. And for those cases where it does happen, it allows to screen newborns and adults to see if you're a carrier for this disease or you're predisposed for this disease. And when you look at different humans, we're like 99.9% the same, probably even greater than that. And the difference between different humans are in what are called SNPs, SNPs, or single nucleotide polymorphisms. So when you compare genomes of different humans, you usually differ. If you just take a stranger and you compare your genome to theirs, one nucleotide out of every 1,000 or 10,000 are going to be different. So not that big of a difference, but those are called SNPs. Right now, through our looking at humans' genomes, there are about 10 million SNPs that are known. There's probably a lot more out there. And people have tried to say, if you have this SNP, will you get this disease? And that is very hard because we're talking about 1 to 2% more likely. So sure, if you get your genome sequence, somebody could say, well, you're 30% more likely to have this disease, which, it's very hard to, do anything meaningful with that information. So personal genomic medicine, as says there on the slide, that is a new field. People are starting to get trained in that, but it's still very, very early days. So it's not all that practical quite yet. But I believe that is the last slide that we have to cover from Monday. So any questions about anything we have covered up to this point? All right, if not, let me stop share and we'll get up the new PowerPoint here. Before we do that, I do have a message for people. So the Academic Learning Center, a program they started a few years back now, are supplemental instruction leaders, SIs. And what an SI is, it's in graduate school, you would call these people TAs, or teacher assistants. What they do is that they will help teach material for lower level classes, kind of like a tutor, but more than that, because they hold their own sessions and they all sit in on classes sometimes. So when I teach Chem 1 and Chem 2, I'm assigned an SI, and we usually have meetings once a week, once every 2 weeks, to talk about what the students are having trouble with, and what to go over in the actual sessions for the SI. So if you are interested in making money over the semester being an SI, if you have any interest in becoming a teacher or going on to a graduate program, this looks great on a resume when going into a graduate program. Please contact Kat Pleak at this email right here. You can see all of the positions that are we're looking for people. So yes, question.
Okay, great. If you're planning on applying to a master's program at MUSA, but it's a biology program, can you still be a TA for Chem 1?
So are you talking about you want to you want to apply to it and you want that to look good on your application. Are you asking like, will they still look at that favorably?
Well, like, would I still be able to like be assistant even though my degree is just biology and not chemistry or biochemistry?
Yeah, it should. So, especially like if I'm understanding the question right, especially here, we don't have a chemistry degree at all.
Right, yeah.
So,
It's tough.
so we get biology students to do that all the time. Yep. So that's perfectly fine.
Yay, okay, thanks.
Yep. And I will post this as well and in our Blackboard in case you want the information there. But yeah, with that out of the way, let's go to our PowerPoint that I had for today. And all the way back. So the one on Blackboard originally I had like February 3rd, and then I noticed that yesterday and I changed it, but then I made the date wrong still. I made it June 15th. Yeah, the date should be wrong on your Blackboard version, but it is the 16th today. And we're going to continue talking about DNA, and we're going to talk about how we can manipulate DNA to do research. And we're going to talk about recombinant DNA technology, also called molecular cloning, also called genetic engineering, also called GMOs, genetically modified organisms. So we're going to talk about how you make GMOs inside the lab. And we're going to talk about the different phases of isolating, amplifying, and modifying DNA sequences. As listed here, 4 different steps to do this. 1, you need some DNA, right? 2, you put that DNA into what's called a vector. 3, you put that vector into a cell. 4, you grow only the cells that have that vector. So we're going to go over each step of this process. So the first thing we're going to talk about is our vector. So this is called a vector or a plasmid, right? You'll see it either way. This is called PUC-18, University, or rather Plasmid, University of California, variant 18, PUC-18. This is not the only plasmid in the world. There are lots and lots of different plasmids out there. This is just an example. And what a plasmid is it's a circular piece of DNA that's usually found in bacteria or yeast. And it ranges in size, as I have there, 1 to 200 kilobase pairs. For us, there are different parts of a plasmid that are important. Things that are generally important is 1, we want the plasmid to have some kind of resistance to an antibiotic or some other way that we can identify the plasmid. So the idea here is we're going to put foreign DNA into this plasmid. We're then going to try and put this plasmid into bacteria. And we only want bacteria with the plasmid to grow. Therefore, if the plasmid has a resistance to ampicillin, which is an antibiotic, we grow our cells in the presence of ampicillin. Cells without the plasmid die because they don't have resistance to ampicillin. Cells with our plasmid live because they do have ampicillin. This is called a selectable marker because we can select which cells have our plasmid. We also want gene production. We use the LAC system for that. Hopefully you've learned about the lac operon in your biology classes. Basically the idea here is when you have a plasmid and you're putting it into a cell, this plasmid usually contains a protein you want to grow. However, you want to be able to control when you grow that protein. That's what the lac system does. We can turn on protein production growth when we want to. So we need some way to control protein production growth. Restriction endonuclease sites, you need a lot of those because we're going to insert DNA into this plasmid. So we need to be able to cut DNA. You also want your vector to be self-replicating. Once we insert this plasmid into the cell, we want it to be able to grow or rather reproduce by itself. So it will have hundreds to thousands of copies of itself inside the cell. So once we turn it on with the lac operon, we're going to be making a lot of proteins. Eventually, it will take up about half the cell's DNA. And usually, the size of a plasmid, the biggest it can be is about 10 kilobase pairs. Bigger than that, and we would have a hard time putting it into a cell. So it does have a maximum size limit of about 10 kilobase pairs. So that this plasmid, this vector, is going to be how we insert, one way, we insert foreign DNA into a cell. This is our carrier. This is our Trojan horse. And here are just some review. So A, we just talked about what factors make plasmid viable for cloning. So selectable marker, restriction enzyme, restriction sites, less than 10 kilobase pairs. Also operon system to turn on and off. So that's the shorthand of the last slide. We can just ignore B. Sometimes I ask a question where I give you examples of like what to insert where. For time's sake, we're just gonna skip B. But C, I do want to talk about. So back in 2018, I'm not sure if you heard this or not, but there was a scientist in China who worked at an institute that helped couples who are looking to get pregnant but were HIV positive. That is, usually this institute helped if the father was HIV positive and the mother was not, so that they could produce a baby that didn't have HIV. Right. So he worked at this clinic. And one of the things we know about HIV is that you need a protein on your cells for HIV to enter. And there are individuals who have mutations in this protein that make them immune to HIV. And so what this scientist did, without telling the rest of the scientific community, is that he made 3 babies that he introduced the mutations to try and make them HIV resistant. He basically made GMO babies. He made the twins Lulu and Nana, and the third baby I can't remember the name of, which was born like 6 months after Lulu and Nana. This caused a big scandal in the scientific community, as you can imagine, because he was modifying the DNA of humans, of embryos, which, you know, we're still in the early days of modifying DNA. And so when you looked at Lulu and Nana's DNA, the modification was incomplete. Usually you have to knock out like 32 nucleotides for for you to be HIV resistant. And he knocked out like 5 nucleotides in Lulu and like 8 in Nana. So didn't quite work there. Also, the knocking out of genes, when you're doing it on embryo, you have to hit every cell or it won't work. And there are questions whether you hit every cell, because you can't really look at every cell of a person, of a little baby. You can't look at every single cell of their DNA. So there's also issues with that. And third, talking about his work, the same gene that makes you immune to HIV, knocking it out has also been shown to increase your intelligence. So there are questions whether he was actually trying to make super intelligent children. He says he wasn't, but who knows? So this person, he was sentenced to three years in jail and a fine of like $80,000. And the scientific community, for the most part, said, what are you doing? Why are you modifying children? There are other people in the scientific community who said, this guy is a genius. He's paving the way forward. So what I want to ask you all as students in science, your upper level students ready to leave undergrad here soon and go on to the wide world with your new knowledge. What do you think? Do you think it is ethical to create or use GMOs? This seems to be a hot button topic now in society. So I just wondering anybody's opinions. Do you think we should create GMOs? Do you think it's ethical to modify DNA of organisms? Anyone have any thoughts or opinions on that? And if you don't want your opinion to be known, you can for sure just send me a private message as well, and I can just read it out without saying your name. You want to have any opinions one way or another? You don't have to talk about the Lulu and Nana. How do you feel about going into the store and seeing non-organic products? Do you feel more comfortable doing that? Do you think that we should stop meddling with DNA? Anything at all? So no, especially when humans are concerned, I do not mind them in food due to large population. So you're of the opinion, do not modify the DNA of humans, but when it comes to other organisms, not so much of a problem, especially since there could be benefits from doing that. All right. Any other opinions? Anybody have thoughts on this? Agree with that? Okay. Well, let me play devil's advocate then. So cystic fibrosis, as we just talked about at the beginning of class. Cystic fibrosis, like 90% of the cases of cystic fibrosis, which is your lungs basically have a mutation in one protein, one amino acid. And when you have this, your lungs can't produce a sufficient amount of chlorine. And your lungs get infected easily. You have a large mucus buildup. Your life expectancy is really low. And you constantly have to go to the doctors and get treatment. So do you think it's unethical if somebody found out that they're carriers for cystic fibrosis and they want to have a child? And so they conceive and they go to the doctor and the doctor says, yeah, your child has the cystic fibrosis mutation. We can fix it by giving it a shot. That's a GMO. Do you think that's unethical? And we should just say, you know what? We're not going to change that. And let's say this is like 40 years in the future. And so we have a lot of experience doing this and we're fairly confident we know what we're doing. Do you still think we should say, nah, we're not going to change that at all? You have this mutation, you're going to have to live with this mutation. Or do you think, maybe for some circumstances, if it's clear cut what's happening, we can fix that? Any ideas on that little wrinkle? Remember, I do grade for participation in this class as well. So I am looking for, this is one of those times that I am looking for participation where I'm not going to call on people though. So just remember that portion of the class. All right, raising hands. Let me unmute. All right, yes.
Okay, so I get conflicted on the situation because I do agree with Lydia about not messing with like humans. However, in the example you gave, I would say to give it to him. If there's something that could help someone and it is a GMO, I would agree with it. So it's an on the fence situation of what's right and what's wrong and when is it okay to use it.
Right. So you're probably saying like if it's to give somebody like blue eyes and make them 6'5", not so much, but something like curing disease, then you could be more.
Yeah, if it's more helpful in that sense, then yes. But just for appearance wise or just for the heck of it, no. I don't think that's something we need to mess with.
All right, great. Thank you very much. So some other chats. I would say that it's okay as long as the method technique has been tested. Thinking that way, I would say yes, it can be good, but only when we are sure in what we're doing, if we have the good technology to do it. I agree as long as it helps someone with diseases. All right, great, thank you. So another hand, let me do that. Yes.
I was gonna say, I'm not really for it just because you're messing with a body that isn't your own, and even though it's not harmful, you're still making choices for something for someone else. So it gets very tricky very quickly.
Yes, I agree with that statement 100%. It is very, very tricky. And it's one of these questions that, everybody who decides to stay in this field, whatever your path leads from here, if you stay in biology / biochemistry in some degree, or you don't, you're probably going to be hearing about this in the future. So I just like to bring up this discussion as something non-memorization. There's no right or wrong answer to this either. It's just something as a society, we have to just be aware of that it's happening. And science, we're no longer as scientists, we no longer live in the age of scientists do whatever they want. and there's no consequences. Like if you ever took intro psychology and you hear about those experiments in like the 1950s where it's like, yeah, we just did this to these people and we didn't tell them we were doing an experiment because we want to see how the human mind works. You can't do that anymore. And science is really guided on moral issues by the society that we're in. So yeah, it's like I said, there's no right or wrong answer. And thank you for those of you who gave your opinion on this. It's one of those things where I'm conflicted as well, because I can give that cystic fibrosis experiment example and be like, oh yeah, it's probably good there. But on the same token, it's like, well, you are messing with an individual who had no say in this. So can you really say it's okay to do? I feel we just need to consider the long-term effects and test them before going forward with GMOs. I am for it, especially it can possibly help things we haven't found a cure for. Yeah, that's the other thing, especially with Lulu and Nana. We don't know how it's going to affect them long-term. Nobody's ever done that to a human, and this person decided, apparently the parents said they were okay with it, but who knows, right? He decided to do it on 3 kids, and now those kids have to live with it forever. Like with HeLa cells. Yeah, HeLa cells are interesting in that, they come from this person who really didn't give consent to her cells being used. And now every single experiment that you do with cells use HeLa cells, because it's like one of the few human types of cells that we found to regenerate infinitely, because they're basically cancer cells, which raises a lot of problems if you ever see an experiment with HeLa cells, because it is a cancer cell. So if you're trying to understand how human cells work and you use HeLa cells, you always have to put that asterisk in, like, well, we are using cells that are special. Well, human cells are not supposed to be infinite. What else? I think that since this is the early days, this is more controversial, but once we get better with this and is able to be automated, then it will be more accepted. I think it should be more towards preventing diseases. All right, great. Yeah, it is early days, so it's experimenting on humans is very scary. If looking at this from a religious point of view, sorry if this is wrong, this is just a discussion on morals. So definitely religion is a part of it. This is still wrong. If you believe this is a greater power out there and they made us that way for a reason, messing with it, the way we were created is considered wrong. Yes, we use medications to help, but altering ourselves in such a way that the intent is wrong, especially when the person has no idea. Yeah, so you cannot throw out that aspect at all of, and that's one of the main things about GMOs that's usually anti, is that you are messing, and if you're religious, you're messing with something that's divine, more or less, right? So who are we as humans to do such a thing? Who governs the ethics of experiment? What law the Chinese scientists break? So the people who guide ethics and scientists are usually panels of experts in their field, who say that you probably should not experiment on humans. Let me actually. So I don't know what law in China was broken, because Chinese law, I am not an expert on that. It just says, going from Wikipedia, like his actual fine. Yeah, he was just detained and fined, actually $430,000. So knowing China, they probably fined him because he embarrassed China in pushing forward, but who knows? Maybe behind the scenes they were giving him a pat on the back. It's hard for me to say what China's intentions were. I say if there's a chance in helping chronic disease that creates a hindering future, I say take the chance, because there's many medical issues that no one know how to help anyways. There's no definite that a person would have other mutations regarding other parts of the body. Yeah, so take a chance. If it helps in chronic diseases, it might help out. And so that's kind of where human modification of DNA kind of is now, where usually if you're going to modify something, it's going to be in adults who give consent to that, right? And you aren't allowed to really touch the germline. It's only somatic cells. I definitely feel like there should be limitations, or else the military could use them to produce stronger, bigger soldiers, or with sports, make them taller individuals could get out of hand. So that is the basics of Brave New World. If you've ever read Brave New World, it's basically a story of where classes are based on their DNA. The top rank people have their DNA all mutated, so they're all like big, strong, blue-eyed, blonde. All the lower class folks are like not that, because they didn't have their DNA modified. So yeah, that is one of those scary scenarios. I don't think it's like that, but I agree with Macy. I could get out of hand. Yep. So slippery slope. And thank you for all the discussion. Hopefully got you thinking a little bit. Again, something we have to wrestle with moving into the future. But with that, let's move away. And also, if you're interested in this scientist, I didn't want to butcher his name because I don't speak Chinese. He Jiankyu, I think is, that's probably not right at all. But this person's name is He Jiankyu. Or I think, yeah, Jiankui, not quite sure. But yeah, feel free to look him up. A couple books have been written on him now. And the kids were Lulu and Nana. Be very, from my research on it just a couple days ago, there has not been any updates on Lulu and Nana. It'll be very interesting to see what happens in the future with them. All right, well, let's move on now. And let's get back to how in the lab, not using humans, but bacteria, how we make our modified organisms. And here on the left, we have the basic procedure of how to incorporate foreign DNA into a vector. So here we have our vector, here we have our foreign DNA. The first step is to cut both the foreign DNA and the vector with endonucleases. And you want the same endonucleases in the vector and the foreign DNA. So you have the same exact overlaps. Once you make the cut, you combine the foreign DNA and the vector together. And then you seal the foreign DNA in the vector and place them into E. coli. What I'm showing here is another type of selectable marker that does not use antibiotics, but uses coloring. It's called a blue-white screen. Here, we just introduce a chemical, and whoever has the vector turns blue, who doesn't turns white. And so you just pick the blue colonies as the ones with the vectors. Now, usually when we grow eukaryotic proteins, we choose E. coli as our system. Can anyone think of what problems we could incur if we grow eukaryotic proteins in E. coli? Or another way to put that is, what are the differences between eukaryotic proteins and prokaryotic proteins? What complications are there? Is this similar to our questions we have for the reading guide? Probably, yes. Are eukaryotes prokaryotes? No, eukaryotes and prokaryotes are different organisms. And so their proteins are made a little bit differently. Oh, okay, mistype. Prokaryotes does not have introns. Correct. No introns. So when you insert your foreign DNA, it can only have exons because prokaryotes don't have the machinery to get rid of introns. One other big thing, post-translational modifications. Yep, as Ju just pointed out, no post-translational modifications, sugars, right? Yeah, so we're going to talk about this when we get to the sugar chapter. Eukaryotes, our proteins, especially mammals, are covered with sugar. We glycosylate a lot of different proteins. Prokaryotes never do that. And so when you're growing a protein inside of a prokaryote, you have to be aware that it's not going to be glycosylated. Modifications. So those are like two big problems. Another one, exclusion bodies. If you grow foreign protein in a cell, a lot of the times, or what can happen some of the times, is that that cell will just clump all those proteins together and they'll become unfolded and useless. So that's another potential issue. So just some things to be aware about when you are growing a eukaryotic protein in a prokaryote. What problems could you foresee with using an endonuclease that produce blunt ends? So I will answer that one. So here we have sticky ends. And let me erase it like that. So this would be blunt ends. So if you don't have any overhang, there's really no reason for this piece of DNA to go in this vector. So you will have low levels of insertion of your foreign DNA, and your DNA could be inserted the opposite way. All right, so let me just erase the markings I made. So with DNA being cut with sticky ends, there's orientation, right? The green has to go with this darker purple. This blue has to go with this lighter purple. Sorry, I don't do colors, so darker purple and lighter purple. If it's a blunt end, there's no orientation that's being enforced by hydrogen bonds. So 50% of the time, it will insert the opposite way, and you'll make the wrong protein. So when you are inserting DNA, you really, really want to do sticky. Blunt would be your last resource. If for whatever reason that it was not working, you could try a blunt end endonuclease. But you always, 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 if you can do it, when inserting DNA, you want sticky ends. In C, I talked about the last thing we're going to do is join these DNAs. That is actually given right here. The molecule that joins DNAs together, that is called a ligase. So that is called ligation, where we are taking foreign DNA, cutting it, and ligating it into a vector. And the enzyme that does that is called a ligase. And for this foreign DNA, one of the questions is, where do you get it from? If I want to study a certain protein, how do I find the DNA of this protein? One, you could actually get the DNA from the organism like directly. That's kind of hard to do, but it is possible. The other way is through what's called a DNA library. So a DNA library is a real thing. It's not just like a database. It's a place that probably has a lot of freezers that contains all DNA fragments of a specific organism. You can Google that. You can like Google like yeast DNA library or human DNA library and go and order the DNA sequence of like any protein that you want. So one of the ways that you can get DNA of a protein you want to study is just buy it from a DNA library. And there's two types of common DNA libraries. One's a genomic library, one's a cDNA library. A genomic library is you just take the genome of an organism and you chop it up and you store all that information. While a cDNA library is you take RNA, mRNA from a cell, and you do reverse transcriptase, and you make DNA from mRNA. And that's how you get your DNA strands. All right, so that's a cDNA library versus a genomic library. And so the question I have here is, if you look at a genome library versus cDNA library, do you think they're the same size or do you think they're different? Based on what I just said, do you think they're the same or do you think they're different? Let's see, how about, I don't think I've heard from this person yet. Samantha Vargas, do you think, based on the information I just presented, do you think a genomic library and a cDNA library are the same size, or do you think they are different? You think there are different sizes. Why? That is correct. Why would they be different? What is a cDNA made of, library made of, and what's a genomic library made of? Well, remember, genomic is everything. And we just learned about at the beginning of today, if we're looking at humans, only 1.5% of DNA of humans is made out of proteins. And a cDNA library is made from mRNAs. So just think about humans. A cDNA would be 1.5% of the genome. A genomic library would be 100% of the genome. So very different. And then B here, would you expect cDNA libraries from different cell types to be the same or different? Why? So I'll go ahead and answer that one for us. Different, because cells produce different proteins, right? Like a skin cell and a blood cell are going to make quite different proteins. So for cDNA libraries, you not only have to be concerned about what organism you get it from, but you also have to be aware of what cell type you are getting your DNA from. Yep, that is correct. All right, so that is how we can get our DNA to insert into a vector. And once we ligate our DNA into a vector, we generally want to multiply that vector a lot. And the way we can do this is through PCR. PCR is one of those techniques that has changed the way that we do science in biochemistry. Made or developed in 1980s by Kary Mullis, one of the most interesting scientists I have read about, in that he worked at a company that the whole company was to basically do PCR before PCR was a technique. It was just to multiply DNA. And he was a big fan of LSD, and he was a big fan of driving 2 to 3 hours every weekend to go to his cabin in California. And so he says, and he says this in his Nobel Prize acceptance speech, which is hilarious, that he came up with the idea of a PCR while on LSD driving around California at night, and just thought of it like, oh yeah, that's how it works. That being said, please don't do LSD thinking you'll get great ideas, because he also says that AIDS is not a thing, and he also says that he's been visited by green-eyed alien raccoons. So he has some interesting ideas, but this is probably the best idea he's ever come up with, PCR. And the idea here is you have target DNA that you want to multiply, and you add primers to flank the sequence you want to multiply. So you need a primer on each strand. You then add nucleotides. That's what DNTPs are, DNA nucleotide triphosphates. And then you add a polymerase and you extend the DNA. So here it is with a gene and a plasmid, so circular. And the cool thing about PCR is that you can also use it to introduce mutations. The way we're doing it here is that our primer does not match our target 100%. You can see we have a mutation at this C, we have a mutation at this GT. When we go and multiply, run PCR, we're going to incorporate a C and a GT, effectively changing that gene. And that is used in biochemistry all the time. If you discover a protein and you want to know how it works, one of the tests that you do is you mutate certain amino acids and you see, does my protein still work? If the protein no longer works, then the amino acid was important. And you do that through PCR. And the steps of PCR are really simple. Add primers, heat it up to 95 to separate, cool it to 55 so your primers will attach. Then you raise it to 72 for your polymerase to work. And you just repeat these steps about 30 times. Heating, cooling, and then heating again. And I have a very low-tech movie where we're going to watch. And by movie, I just mean a bunch of slides in a row. So we start with our DNA sequence that we want to amplify. We heat it to 95 to separate our double-stranded DNA and the single-stranded DNA. We add our primers at 55. We extend at 72. That's the end of round 1. In total, we've made 2 DNAs. Star DNA, that's the DNA that we want. Non-star DNA is the DNA that we don't want. And by what I mean by want is we're just trying to amplify one sequence, right? So we're just trying to amplify this part of the DNA that I drew lines across. So we only want DNA that is the boundary of that length. After 1 round, we have not done that. We have multiplied our DNA, but we do not have the correct size of DNA. That moves up to round 2. Heat to separate, cool down to anneal, go, raise temperature to extend. Round 2, we have made 4 DNAs. None of them are the DNA we want though. So 4 non-star DNAs, 0 star DNAs. So round 3, heat, anneal, extend. Round 3, we have finally created 2 DNAs that are star DNAs. So these DNA sequences are the exact length that we're trying to create. So after 3 rounds, we have made 8 total DNA molecules. 6 of those are not the correct length that we want. 2 of those are. So that's round 3. Round 4, heat, anneal, extend. All right, now we're getting somewhere. Round 4, we have made 16 DNAs. Half of those are DNAs we don't want. Half of those are our sequence that we want. So right now we're at 50-50. And I'm not going to run through all those cycles like that. Instead, give you the table. Is that if we look at the very end, after 10 rounds, we have made 1,024 copies of our DNA. 20 of those are DNAs we don't want. 1,004 of those are the DNA sequences that we do want. So with this technique, if you're just looking to amplify one specific sequence of DNA, you run it for 30 times, you're going to have millions and millions of copies of that sequence that you want. For a plasmid, all the copies would be good because it goes in a circle, right? So you just copy the whole plasmid. So if this was just a plasmid we're looking for, we don't have to worry about star and non-star. So after 10 rounds of multiplying a plasmid, we would have 1,024 copies of that plasmid. You can see this is just exponential growth. Like 30 rounds, you're just going to have millions of copies of a plasmid. Right. So before we move on here, any questions about PCR or anything we have covered up to this point? All right. So some PCR questions, and I didn't go over ideal primer characteristics. So let me go over that. So for those primers we were talking about, what do we want out of these primers? Generally, you want these primers to be like 18 to 20 nucleotides long. you want 40 to 60% GC content. You want the last residue to be a G or C generally. You want the melting temperatures of your primers to be within 5 degrees of each other, 5 degrees centigrade of each other. Because remember, you have to add 2 primers. So they have to have around the same melting temperatures. And you want their melting temperatures to be like 65 to like 75, somewhere around there. Let me just double check that I got that temperature right, because I'm doing that off the top of my head here. So let me just quickly check my notes. Yep, 65 to 75 melting temperature. And so the next question, though, is some things that will help you better understand PCR if you understand the answer to these questions. And so let's talk about linear DNA. So I have linear DNA that I'm trying to copy. And I only add 1 primer. What is the outcome of doing PCR with only adding 1 primer? What will happen during my experiment if I don't add both primers? Only 1 strand gets copied, will only replicate 1 strand. Yep, correct. All you're going to do if you just have 1 primer is keep making this strand over and over and over and over again, right? So you'll have incomplete DNA. You'll only make half DNA. You're going to get a lot of single-stranded DNA. All right. So let's go to our next situation. Our next situation is we had both primers, but one of our primers sticks to multiple locations. What's the outcome of that? We would get incompleted or mutated DNA. Not so much mutated. You would definitely get incomplete DNA because you would like extend here and then like stop, then extend here and then like stop, then extend here and then you would stop at the next primer. So you get very short, choppy DNA sequences. So that, and you would always have like a gap here because we don't have the machinery to fill in that gap. Would it be more than 1 copy is made of that strand? You would copy that strand correctly. It'll just be in multiple pieces because that primer that's like that I drew, like, so imagine that we are going this way and there's another primer right here. Your machinery for making new DNA once it hits that primer site, it's going to fall off, right? There's already DNA there, so it can't get past that. So it's just going to stop what it's doing, and you're going to have this gap here. Inside the body, the eukaryotes, this is actually how we replicate our DNA. We have a lot of different start sites, and we also have Okazaki fragments that are basically this. What we're talking about is we're making Okazaki fragments more or less, but we don't have the correct machinery to combine our fragments together. So you'll get one strand that's very fragmented. So we never have like complete DNA, complete double-stranded DNA. All right. Next example. I have DNA. But I have a single-strand break. What would happen? There would be a deposition of energy. Explain what you mean by that. The total DNA will not be copied. So that is the answer that I think that people generally give, that DNA will not be copied. However, this one's tricky because let's actually do one round of PCR. So red primer goes here, and we copy this fragment. Blue primer does this, and then it stops because there's a gap. However, remember DNA, each strand's a template. So this red strand is a perfect copy now of this original black strand with the gap filled in. So you would actually be able to copy your DNA perfectly if you just have a single strand break. Because as long as the other strand is not broken, you will make a copy of this strand without a break once you do one round of PCR. So no problems. The problem comes when you have a double-stranded break. So let me answer. I'll just do that one. If you have a double-stranded break at the same point, there's no way to fill this in, right? You don't have any template that can fix this. So you're basically going to have no reaction. You're going to have incomplete DNA being made. Inside the body, you have to do some tricky processes to fix a double-strand break, where you have to have like your other chromosome come in and share some of its DNA, doing overlap, strand exchange, and all that. Inside a PCR reaction, a double-stranded break means no reaction. Why would there be a break in DNA strands? Even though you said never mind, let me answer that. Inside your cell, inside of every one of your cells, you have about 30 to 40 double-strand breaks a day, and you have a lot of single-strand breaks. DNA breaks all the time. So for like your template, if you like shake your tube, you'll cause DNA to be broken. So it will just break. DNA is a lot more fragile. We have a lot of processes to fix it, but inside the cell, it breaks and mutates all the time. If you're interested in that biochemistry too, we talk all about the processes to fix broken DNA. All right. Any questions dealing with PCR at all here? All right. That ends chapter 3. And we'll move on to chapter 4, which is our next biomolecule, the amino acids. My favorite biomolecule, amino acids and proteins. And all amino acids have this general structure where they have a carbon alpha, and they, again, I got this off the off your book, so I hate when books and the internet do this, but it should have an amino group that's like gonna be charged under normal conditions, so NH3 plus, and it's gonna have a C terminal group, COO minus. This R means anything. And inside the body, there are 20 different R groups that are common. Some people will say 21, but most people agree it's 20. Those are the standard amino acids. And you'll have a hydrogen. Amino acids are what are called Zwitter ions. Those of you who have taken German or know German, Zwei means two in German. So this is 2-ion compound, which it does, has a positive ion and a negative ion, or cation and anion. For biochemistry, and in the videos that I had you watch before class, I made a fool of myself trying to tell you ways to remember the different amino acids. Like, what's a pirate's favorite amino acid? That's arginine. Or who's the cutest amino acid? That's glutamine, because the one letter code of glutamine is Q. So your task as a biochemistry student, and every single person who takes biochemistry has to do this, and it's painful, it's no fun, but amino acids are the alphabet of proteins. And if we want to talk about actual proteins, we have to know what they're made out of. So we need to know the full name of all of our 20 amino acids. We have to know the 3-letter codes of our amino acids. We have to know the 1-letter codes of our amino acids. We have to know what our amino acids look like. We have to know which side chains have to are ionizable, the pKa of our side chains. And we just have to know that this N terminus has a pKa of 9.4 and the C terminus has a pKa of 2.2. In reality, each amino acid has a different pKa for N term and C term. So I'm just going to say they're all 9.4 and 2.2 for simplicity. What do I mean about pKa? pKa, if you remember when we talked about acids and bases, it's the acid dissociation constant. And generally, what the pKa means is that if your pH is lower than your pKa, you'll be protonated, right? So inside the cell, the pH of the cell is 7.2. And the N terminus, the pK is 9.4. At a pH of 7.2, it is protonated. C terminus, right, pK of 2.2, we're above that, so it's not protonated. That's why I added a hydrogen and took away a hydrogen. I will post a sheet that has all this information for you. Feel free to Google like amino acid flashcards. Like anybody who wants to take the MCAT, anybody who ever takes biochemistry has to learn this. There are a ton of resources out there. So don't feel free that don't feel the need that you have to make like your own flashcards or make up your own mnemonics if you don't want to. Just look online, you'll find a ton of information. But we do need to know this information. And we're not really interested in amino acids by themselves. We really care about amino acids in a protein. And so here is a reaction where we are forming what's called the peptide bond. So we have amino acid 1 and amino acid 2. We take 1 oxygen and 2 hydrogens away, and we form what's called the peptide bond. Now we are making water in this reaction. And I think I said this in the video, but what is that reaction called when your product is water? The forming of a peptide bond is a what type of reaction? Condensation, correct. Because we're making water, so we're condensing water. What's the reverse reaction called? If I want to cut this peptide bond, the arrow for water would go the other way. What is that reaction called again? Hydrolysis, correct. Anytime you see the word lyse, that means cut. So hydrolyse means water cut or hydrolysis. Right. So condensation makes a peptide bond, hydrolysis cuts the peptide bond. And one amino acid, tyrosine shown here. Now, if I just have pure tyrosine, that is tyrosine not in a peptide bond, tyrosine can be soluble in water. Barely, but it is. So one question I have for people is, let's say I have like 5 tyrosines and I put them in a peptide bond, right? So all 5 tyrosines are in a peptide bond. Do you think this polypeptide chain of tyrosine, therefore, would be more soluble in water or less soluble? So the question is single tyrosine versus multiple tyrosines in peptide bond. Which one is more soluble? And actually, I didn't have a poll for this, but let me use one of my old polls. Let me just see which one's good. All right, I'm going to use the question timer for this. So the way we're going to do this is if you think the single tyrosine is more soluble, answered finished. If you think the multiple tyrosine is more soluble, say I got partway through. I know it's kind of weird, but if you think single tyrosine is more soluble, answered finished. If you think multiple tyrosine is more soluble, answer I got partway and then not sure. Because making polls on the fly is not easy in Zoom. So we're going to use this poll. All right, so we have answers here. Can we go over reading guide 3, question 10? It's related to the slide. Sure. Let me just answer this and I'll pull up the reading guide 3, question 10. So the majority of people think, and it's not a big majority, but the majority of people think that multiple tyrosines is more soluble than single tyrosine. And the correct answer is single tyrosine is much more soluble than multiple tyrosines. Let me erase all this writing so we can take a look at why that is. So remember, if you're an amino acid, you're a Zwitter ion. You have a positive and a negative group. Tyrosine looks like this. Has a big phenol ring, a little bit OH. So when you stick this big group onto this R, the N-terminus and C-terminus is what is making it soluble. In a peptide bond, you are losing your charges. You're still polar, but you're no longer charged. So if I have multiple tyrosines, I still only have one charged positive terminus and one charged negative terminus. So you, by combining amino acids, you are losing charge. Therefore, since tyrosine is only barely soluble to begin with, if you have a long peptide chain of tyrosines, that's not going to be soluble because the main interaction are going to be these phenol rings that are copied. And these phenol rings aren't soluble by themselves. So less soluble. Does that make sense? Or people still unsure about that while I pull up the reading guide, which we had a correct question on. Question. Was it reading guide 3, question 10? Because looking at the one I just pulled up, question 10 has to do with what's the difference between a haploid and diploid organism. Maybe I'm looking at the wrong, did I post the wrong one? One sec. I'm gonna stop sharing here so I can go to my actual course site. 1, 2, 3. Oh, yeah, I guess my reading guide 3. Okay. Yep, I was just looking at the wrong folder then. All right, draw a cis, gly, and asparagine tripeptide. I identify the peptide bond and I was supposed to say the N and C termini and determine the peptide's net charge at a pH of 7. So instead of so let me just look at where we're at. Yeah, so we're actually going to do that next. So instead of like actually doing the reading guide one, let's do the one I had in the PowerPoint. Because the next question is draw the peptide tapes, right? So what I would like you to do is find some chart of amino acids And I can do that as well. And I want you to practice drawing the pentapeptide that has the sequence TAPES. Let me pull up let me see if I can just get a chart here fast on the internet. I'm going to cancel this and pull it back up once I make this change. That one, this one. Yeah, let me reshare that. All right, so now we have a chart of amino acids. So see if you can draw TAPES in a correct peptide. Right, so I'm sure you probably did not have enough time to draw it all. But let me just show you my bad drawing of it. And I'm gonna remove the amino acid thing so you can see my drawing a little bit better. All right, so I'm not sharing. It would be helpful if I shared it. All right, so this is the pentapeptide TAPES. So I forgot to fill in the H's here. So some things to note is that all peptides begin with an N terminus and they end with a C terminus. And we always write it like that. It's always N to C. Connecting every single amino acid is our peptide bond. And that's what I'm blocking off right here. So this peptide bond is showing us where one amino acid ends and then another one starts. So this is T, A, P, E, S. One other thing to note is every peptide bond looks the same except peptide bonds that have proline in it. When you look at the side chain of proline, you will see that it is the only amino acid that will bend back on itself and bind its own nitrogen. What that means is that any peptide bond that has proline in it is missing the hydrogen from the nitrogen. And that's very important when we talk about protein structure. So that's just something to keep in mind moving on when we get to like chapter 5 and talk about protein structure. Some other things that I want to point out. What I am circling in red, minus the proline, I kind of got overzealous there. These are called backbone atoms. So the atoms, the N, the C alpha, and the carbonyl, those are what we call backbone atoms. And then all the R groups that I'm the circle here in blue, these are called side chain atoms. So when we talk about structure, we're going to talk about backbones and we're going to talk about side chains. And we're going to continue this discussion on Monday. So we have a few more things that we need to finish out, mostly just talking about charges of peptides. So we'll finish that on Monday and then continue our discussion about amino acids. So before I let everybody go, are there any questions about anything we have covered up to this point? All right, if there's no questions, will the oxygen left ever be in enol form? Usually the way I have drawn it is how it's going to be most of the time, like the vast majority of the time. It is possible that it's going to be an enol, but very, very unlikely. So unlikely that we don't consider it, but that does not mean it's impossible. You know, one time out of 1,000 maybe. All right. With that, thank you for participating. I appreciate it. Thank you for spending your morning with me. have a good weekend. If you have any questions, please let me know. I'll put a homework up and I will see you Monday. Yeah, take care, everybody.